So starting recording now. So we need to identify, and we all know that some proteins contain some of the conserved structure, conserved sequences, and having particular conserved region, so that the protein remains stable in most of the organisms. So those are some of the patterns you can say, or the sequences that gives rise to different kind of functions. This can be characterized into two things. One is this motif, another is the domain. So what is the motif is that short conserved sequence pattern that is associated with different functions of a protein or DNA. That is the domain is conserved sequence pattern that is having independent protein structure and function. So if you can look at it, that this is how a conserved region of sequence can be denoted, where E followed by unknown any region any amino acid sequences, which should be given by any two unknown sequences. Then followed by either of S, H, or M. So if this third bracket is there, so it represents that either of these amino acid sequences is there. Again followed by any four amino acid sequences, and this bracket, second bracket, means that it can any amino acid sequence can be present, excluding this. So excluding P, any amino acid can be present, followed by L. So this is a representation of one kind of a domain. So this is how generally uh, domain sequences are represented as. Now let us look at some of the sequence pattern databases. So if you go at ProSite, so you all can go to ProSite, search here. Just you all are able to follow, right? So you can see this database where it is mentioned that it contains of the database of the protein domains, families, and functional sites. Okay, so it contains everything, the different types of functional sites, associated patterns. So if we go and search for this, you can know if some knows the zinc finger and all are there. So just we go and search this SH3. So for this, you can find that for this expression, this amount of things are there. Accordingly, for your sequence for your particular expression, you would search. If you go, you will find that this is the contains the description. Also, you can go with this ID if you identify. So it will contain the description of what it does. Uh, this domain contains of how many amino acids. What are the other things, functions that it follows, where it has been last updated, and the technical section is from where it has been retrieved, and the references that which papers have reported about this particular domain. So this all information you will get at a place. So you can go by just looking at each section, whichever section you want or what information you want, you can go and search for it. So whichever domain you are curious about or you want to know, you can all search. So ProSite contains a extensive database of all those domain and protein families. Now, if we look at another, is that the of the particular domain database. Okay, so we can go and search prodome. So you can go to Prodome also. So see, the Prodome is if you go to the whole database, so display a Prodome all proteins in Prodome families. You can get by the search by the related databases, or you need not heavy entry or keyword search. So for that you need an entry of an accession number. So that accession number we will have to get it from GenBank, NCBI or anything, ID entry term. Again, which compare your sequence with Prodome. So any sequence identification or pattern identification. So you just imagine how you will try to identify. For your identification you need to have the sequence get aligned. So for the alignment or for to identify the regions of the concept sequences, then only with the it will match with the database and give you the result. So if you can use that, you want to have the blast p with the protein sequences, or if you give the nucleotide sequence, then also Prodome can work. It will perform the blast phase by translating it with the protein sequences. And which method? So the multiple alignments or consensus. So consensus will generally depend, does not perform the alignment, and it will look at the uh, sequence only and give, and also the expect value. So you can perform this. So if either you can, so you can paste your sequence also. So let us take one sequence and look for it. So if we try to find out today, nucleotide. So let us take the nucleotide only. We perform the and we'll do the blast take. Expect value wave, okay, and we'll submit it. This is we are doing using blast. Via blast, we are trying to search out the domain families, and other with the accession ID, we can search it. All of you are following it, right? So you can see that it had performed multiple alignments. Program has been NCBI blast text. Matrix that has been used is Blossom 62. So it has their default, they uses Blossom 62. So hopefully you all are now clear, right? What is Blossom 62 and all. So it threshold is 0.01. So now here's a graphical representation from the HSP that the performance found by the blast. Sorted from that so that lower bases may be hidden. So this is what the results have been identified. So the domain position 3 to 77 amino acids. So high scoring segment pairs from there you can see that 3 to 77 has one domain, 54 to 167 has one domain, and their score and that E value. So the closest domain which has been present in their database and with what the sequence it has identified, it has got those databases. So these are all results you can get using performing blast in proton. So now there's another is that PFAM, which is generally gives about the of the protein families. That if a particular protein is of from which family. So, for example, you can find data in PFAM. Here you can go for analyze your protein sequence, or you can view a group of related entries. So you can all find the domains of on a PDB. If you're using a PDB structure, also you can find domains. So if you go for sequence search, so here you have to give that sequence. Okay, that domain. So you can just use this. So here you will get this kind of result where you will get all the length reference. So PSAM, so you can see that domain is CH RoJF. So what is RoJF domain? You will get an idea about knowing that what is the RoJF domain. You can know about this of all your sequences. Correct. So this is RoJF and this is the pH. So for this domain also you can know. So anything. So let us try with our sequence one. So you can get a sequence from Uniprot ID. If we get this expression ID, we have to go to Uniprot to identify the get the expression ID of your required protein. And again, if you give, then so you can see that hemoglobin is the domain will be globin as we know. It has given that subunit gamma 2 or length. So you can go and read more about it structure. what if it is linked with us, all those information you can get, as well as you can know that what is that PFAM ID. Okay, so you can, and bring the pro site, what is the ID that the pro site that is present as the database. So you can get all these results, retrieve these results. So another website is there, web server is there. It's called the Interpro. This much includes uh, all those functions of those which we saw now just now, using two site prodom or QFAM. So Interpro helps immensely in the classification of the protein families. It's a very good tool which can help you in identifying many proteins. So let us search by sequence. Okay. So we will go and just copy paste this sequence. So here you can see that valid sequence it got tick mark because we've given the first sequence. Hopefully you can now appreciate all those sequences and all how it works, because since so many websites, so many servers, so many things have been developed, we need to have a common format in everywhere so that we can exchange the data, and one software can read another and understand and can interpret. So this is the name, job title you can change accordingly. So see, so families, so what I told you that it that all those individual databases that we do, Interpro has integrated all in it itself. So you can identify family, domain, sites, repeats from CSFLD, Panther, MAP, ProSite profiles, ProSite patterns, Smarts, PDT, prints, PFM, PIRSF. So we don't need to go to individually to look at it, and if we just check it in this Interpro, we can get all those results compiled at a go. And otherwise, if we are specific that we want to check from this particular website or web server only, then we can go and select that accordingly. So structural domains websites are they are using superfamily Cathegin 3D. So these, and you all can go and check these individual websites as well, how they function and how. So mainly all these tools generally based on two or three methods generally. That is, the one is the ab initio based method, which is just check on the sequence part, and another are those which based on the modeling and the alignment part. So you all can check out all these routine this website that which function they use, and accordingly according to your uses you can perform. So now we are keeping all this, and now we will try to identify. So TMHMM is that which uses hidden Markov model. So let us search now. So now it is searching. If you all have any question, you will can ask. So if you can see that it takes a little time, as it has all those software, it will search all those things as we have. check all those websites and web servers. So it will go through all this and then will give the final result. So it generally takes a little time, but it is a very useful tool as you can don't have to go everywhere and just with a one you can get all those results. So if you identify any, in fact, unknown protein or any unknown sequence, and it belongs to which protein family or what are those domains, you can easily identify through this tool, and you don't have to much worry about. So it got finished. Just look at the results. So overview, you can see that it will see all those tools will give you that what have you submitted or what is the length of the amino acid. This was the selection. So here you can see that it could not predict any protein family. So it could not predict any protein family. So it has given none predicted, and also so that's why it did not give any geo terms as well. But this should not have been the case. But because it's an mRNA, I guess it could not predict any protein family. If I show you here one example, see here for one another example sequence, you can see that it could identify protein family membership as pyruvate flavodiexin oxidoreductase family. It belongs to that particular family. So it gave that family its IDs, homologous superfamily, conserved site. You can get that which site is conserved and basis of what it has predicted and everything and the predictions. The geo terms is the gene ontology terms. So any protein, so three things are generally required to understand the full function and the structure of the full utility of that particular protein is that biological process where it functions like electron transport chain. It helps in electron transport chain. What is its molecular function? So its molecular function is oxidoreductase activity. So it helps. It might. These are all predicted things. So it might be working as an performing during the oxidation reduction or iron-iron binding. These are all predictions based on the source, catalytic activity or oxidoreductase activity, and the cellular component is it could not predict the cellular component means that where it is localized. The protein is localized at which place, subcellular location. Is it in cytosol or nucleus or ribosome or mitochondria where it is? So it could not predict, but it generally comes. So to understand any protein, these three things are generally needed. So for this so for any protein sequence you have and you want to know which is not present in the literature about the protein and you want to identify. So if you just come in Interpro and if you just give, you can identify all these things. There's protein family membership, geoterms, and everything, and you can get an idea that what protein, what this protein generally does. So, so this is how you can get. Okay, so basis on this, you can identify the protein motifs and domain. So now another is that protein family databases. Right, so see, so another databases is there that is based on the phylogenetic classification. Let's look at that database. The databases of clusters of orthologous genes. This is included in the NCBI thing. So COG stands for clusters of orthologous genes. You can see that when it was database was created, that was created way back in 1997, and most recently it was updated in 2014. So it includes complete genomes of 11X1 bacteria archaea that move into. So generally for bacteria archaea it includes, and so the thing is, the concept is that there will be some orthologous or the conserved genes that performs the cluster. So all those evolutionary traits over the evolutionary trait, some clusters of genes shall remain conserved all over the time. So with all this you can find out. So if you just try to find out using this, you can use protein name also from the NCBI. So we'll look that also. So see, this COG PDB pathways, which pathway it is working. This is nucleoside diphosphate kinase. The organisms it can present in, it can identify from where. So let us look at our protein sequence.
Excuse me, sir, I have a question.
Yeah,
So while using the Interpro,
yes,
there was a term called when the protein family membership none predicted, and also
yeah.
there came a message that go terms, no go terms. Like what is this go terms?
So Go terms is this. This is a gene ontology term. So as I see this, according to the based on the protein family, it predicts, right? It predicts that it what will be its that molecular function. So generally, it is known that these kind of proteins are present for a particular help in performing a particular cellular function, particular molecular function, right? About particular cellular processes like electron transport chain or glycolysis or like those. So biological processes. So here you can see that it predicts that it performs helps in electron transport chain. Another, it helps in what is its particular function, what in what step of the reaction it's help. So it can help in oxidoreductive activity or iron binding or catalytic activity. Another is the cellular component, which at which part it gets localized or it is present. Okay, so these are the geo terms. These three things generally, whenever you work with an unknown protein sequence or get identified with anything, or you want to know from what is its actual function and thing, so you need to predict and identify these three terms. So it's clear.
Yes, sir.
Okay. So we'll yeah. So it is like this. waiting for a protein sequence. You can see meanwhile you can see that these are those categories. So for all these, they have these clusters of the gene sets: RNA processing, transcription, replication, extracellular structures, lipid transport, and all these. Also, there are pathways. So these are all the pathways and the number of COGs. So, if, for instance, if you want to know about particular aromatic amino acid biosynthesis, and you want to know how many COGs are there, which are there, you will can come and search here. Okay, so if you want to get all the COGs, so you can with this API format also you can download all those COGs if you want. Also, you can find filter COGs if you know filter COGs by organism name for particular organism which are those COGs. So you can do perform all these steps here using COG. Now another is that motif that you can see that in these databases, that is this prodome or these domains and prosite, it contains the databases. But if you have a protein sequence and you want to identify, so how will you identify the motifs for an unknown protein structure, unknown protein sequence? So that can be performed through here. So yes, you can if you go here the meme source, so you can see that motif-based sequence analysis tools. So here you will give your DNA, RNA, or protein sequences. You will get the motif discovery. If you give through the motif databases, this tool gets used, have the enrichment of the motifs, and you have the geo databases added to it. So will have the discovered motifs, enriched motifs, annotated motifs, and hence why you can scan for it and identify it. So, so if you go and hover over all these tools, so you can find that what tool does what. Like here, you can see that BM discovers novel ungapped movement in your sequences. So how does it perform? Mean splits variable length patterns into two or more separate motifs. So what is stream? So based on if you go and read all those, so based on at which tool, particular tool to use, you will use that accordingly. Extreme comprehensive motive analysis on sequences can be anywhere in the sequence. So if you don't know that at which particular place it can be. So here the input sequences may be of any length and their length may vary. So if you are not sure of your length and where it is be present, so it can be present on anywhere. Then stream discovers ungapped motifs enriched in your sequences. So LAMP to gap motifs. So if your in your DNA has gap sequences are there, then how to you will need to use this one. So short ungapped motifs. So you need to use this tool. So motif discovery tools can be followed by this. Then motif enrichment can be performed using this. So that's how you can perform. So let us try it once. So classic mode DNA or RNA protein. Input the primary sequence. So you can to type in sequences. RNA, yeah, RNA. Yeah, so we are not giving any that how many motifs has to be distributed in sequence as we so if you can give one occurrence per sequence or any number of repeaters, if you know that it's okay, how many motifs should we find? You can add that how many motifs it should find, and then start search. It will give that your main job is now waiting. So one DNA sequence. So one DNA sequence you have given. So now we can go for our output. So see, you can see that these are the discovered motifs. You can have seen, might have seen like these things if you have gone through some papers and all. So these are the logo. So if you go and click the sequence logo of the motif, the rules for construction description section, you can go and search that how the motifs have been given. Okay, so if you go and search, so there you will get all those parameters and the behavior examples, take logo, how it is given and all. So now you can see that the E value. So what is this E value? The statistical significance. So each E value generally was the statistical significance of any particular sequence or anything that has been predicted with low E value motifs first. So then the sites, the number of sites contributing the construction of the particular motif, width of the motif, okay, the motif location, where it is present at the starting site, end site, so input sequences, everything. So here, this is how you can predict for your motif using using this tool. So this is how expectation minimization algorithm is work behind identification of all these motifs in this particular tool. So this is how you can identify the protein families using COG for the cluster databases. So this is justly an orthologous case, and not only not does not predict anything, but motif for identifying protein families you can identify through either PFAM, domains through prodome and prosite. Also, you can have interpro where it all those things are clustered at one place, and at one go you can know the about your proteins, given protein sequence. Here you can identify motifs using mean suit, using multiple EM for motif elicitation. Now we will come to the gene and promoter prediction. So now if the protein, if what we also know that if your DNA sequence is given, we have to functionally annotate those genes and genomes, and what should be the, what are the genes that will be the protein coding regions or the functional RNA coding regions. So if we are DNA sequences are not known, or if we identify any that sequence of DNA, we need to identify. So there are many tools that helps us in identification of uh, gene regions, as well as for the promoter regions in the for the regulatory elements in the prokaryotes. So the thing is that the identification of gene also again works on identify using consensus pattern by looking at the sequence, or when the tools are trained by looking at the number of sequences, because all those gene regions have contains particular elements, like starting codon region of the sequence generally starts from ATG or AUG that codes for the uh, methionine, as well also polyethyl. Also, there are some the sequences that demand conserved, like some Tata box and all other sequences that are generally responsible for the regulatory elements, and those starts from specific sequences. So we can easily, the computational tools can predict the gene regions. So let us look at one tools, how it is does. We will go to gene mark. Sorry. So if you go for gene mark gene production prediction. So if you go to this tool, So there will be gene prediction in bacteria, archaea, metagenomes, gene prediction in eukaryotes. The gene prediction in bacteria, prokaryotes, and eukaryotes will be slightly different. Also, gene prediction in transcripts. So for that, gene mark S is there. For eukaryote, gene mark ES is there. For bacteria, gene mark is there. So if we go to identify this, so we need to perform for prokaryotes. Right. So if we go at CBI, if we go for nucleotide, and if we write pseudomonas, so with this, we would like to identify the gene regions present in these organisms in this DNA sequence. So we will just go enter sequence. So using this output for gene production, output will be gene nucleotide sequence. We need to identify the genes and then we will start. So this is will give the output. So you can see that this is the gene mark which has been run at what time, and it gives the model information that what model has been used, and this according to it. So a gene stands from the stand is positive strand, and left end is starts from 292 and ends at 2088. So upon the 20 to 20 nucleotides from 292 to 2088 is the gene region. So for this organism, you can easily identify that which regions are genes. It could predict the gene length and the. So this is the. So this is the gene length. So you can identify easily. Now, so looking for another is glimmer HMM. So we go. So here you can see that glimmer HMM is a eukaryotic gene finding system. Generalized, it is based on a generalized hidden Markov model. The hidden Markov model is that which Markov states are predicted that generally varies. So although the gene finder, so but we for Glimmer HMM to run, you need to have a Linux system because it is not a web tool, but you need to install it and then only you can run. So just gave you that there is a software like Glimmer HMM also that helps in identify eukaryotic gene finding system. So this is so now another tool is this FGenes. So for FGenes, will let us there is a suite actually where you can find many tools. So this is the company homepage. If you go, so if you can see that you can identify having various things like eukaryotic genome annotation program. So eukaryotic genome annotation pipeline using FGenes H++. So here it will do is mapping known genes or mRNAs from RefSeq or other sources. It will map to your unknown sequences. Then it mapping known proteins to genome, identification of splice sites, identification of gene, then bacterial genome annotation, Then eukaryotic and bacterial genes, functional sites identification and genome annotation. So each contains F gene size, F splice, B prom, prediction of bacterial promoters. So if you go to this software, you will have a full suite of tools available, promoter and functional size prediction that we'll be looking at. So it all can be found here. Using in one go, you can find and you can choose and use any tool. So here we will be looking for F genes H for eukaryote. So F genes H, what is that? The HMM based gene structure finder, most accurate program for plant gene identification. So it is generally helps in the identification of the plant genes. So if it is that, then we can search for Arabidopsis. Let us look at this region. So paste the nucleotide sequence here. So we can give the organism and we will search. The prediction of potential genes. So it did not give any reliable prediction. So this region does not contain any predicted as much genes. So let's try this once that if we can get some predictable results. So here you can see that we here the genes have been predicted. Number of predicted genes have been 1 in positive stand 1, minus in 0. The structure start end, coding sequences, mRNA of 2 exons. So here in the eukaryotic gene prediction, it becomes a little bit more complicated as the introns remains present within the exons, and that computer tool has to keep in mind and to remove the introns and to identify the exons. So it has done that. So it is that this to one mRNA that has been given, right? So this is this. Now if we go, initial is the F prompt, right? So here human promoter prediction. We go at F prompt. So here see in this software suite only, so you can get F prompt. Just one second. So let us check. So this is the human promoter prediction. So let us search for any human sequence. Go for mRNA. So this it contains this all the collection of the tools we can easily identify it. So you can for since is a human thing, so you can give for the data box all threshold and all. So we'll just process it. It will predict the promoter regions in the human. So you have said that the zero promoter and enhancer sequences are predicted. So it does not contain any promoter or enhancer sequences. So if we go and try to site identify another one, so just process it. It also is not predicting, but this is the tool if you find, so you can utilize that. So now another is that, or another tool is that for exponent promoter prediction, we can go there. Is first yes. So this is the tool. So we can just paste the sequence. So cutoff values are all you can give accordingly. The maximum acceptable length is 100 kb. So there might be some issue in the server now, but this is one of the tool which you can use. So now after this, you have another is that CPG island. We know that's the phenomenon of CPG island. Generally, the promoter areas have a lot of these CPG islands. So there is another tool from where you can, with the basis of the concept of CPG island, you can detect promoters. So CPG Pro D. So it is dedicated promoters associated with CPG islands. So whichever the promoters that are associated with similar islands, it will be predicting those. So you can download it as well as CPG island promoter detection. So here you also can humans. So here you can give and submit. But we need to. So here you will was obvious that it won't contain anything because number of CPG islands it has to be found. Means it has to be related to it. Then only it can be able to identify it. Then that G plus C frequency and all in the basis of that promoter. We can try for any promoter genes. Mouse promoter genes. Okay. Unsaturated protein 53. If we go to yeah, 53. So this is from MGI something details. If we go for tier 53. So we try to find out if we can get using CPG Island. So no, so we can find out using this only. So I think these are all what so we can predict the gene region, we can predict the promoter. The software it contains all those tools and protein families and all can be identified through Interpro. So another thing is that there are many chances because of the computational predictions to be having true negative or false negative or false positive. So we need to the two terms that are used for this is the sensitivity and the specificity, which helps us to identify that the corrected gene prediction is correct or not of the accuracy marker. So sensitivity gives where true positive divided by true positive plus false negative, where specificity is that true positive by true positive plus false positive. So this was all today. So if you all have any question, you can ask.
So only one
Yeah,
question that in
yeah,
case of the databases you showed us and demonstrated,
yeah.
is there any kind of databases which predicts or shows in case of a 3D structure or depicts it in a 3D form?
Yeah, so protein sequence, if you go like there, if you want to identify the 3D structure or the 3D form, what protein sequence, if you want to see, there are other software not related to it. Here in one or two tools you can identify, but there are many other methods of forming the structure.
Can you give
Correct.
one
So
example?
if you have a protein sequence now, so every protein structure, if you can go to AlphaFold, you can get a predicted structure for any protein. Okay, like this. You can PDB file you can open. So you can search the for the protein. So now it's become much robust. But here also you can identify some of their where 3D structure visualization was there. You can find out in Interpro also if any there are many tools are there now. So which gives the protein structure and also AlphaFold is there. Another way of predicting structure is homology modeling. ITESR is there. There are many other tools and there which I think we can do it in another session on this protein structure predictions and all. Okay.
Thank you, sir.
Yeah. Any other question? Okay. So if not, then we can end this session today here, and we can resume from next. Thank you all.
Thank you, sir.
Thank you, sir.